And this is really one way we can do it. And by recording it and putting it on our YouTube channel, we're hoping that we can reach out to more people as well. I believe there might be over 50 on this call tonight, or certainly over 50 people have responded. So that's absolutely fantastic to have you all here. Vicky has kindly offered to facilitate the Q&A between Pete, Martin and Lucy's presentations tonight. Each presenter will present for 20 minutes and then 5 minutes Q&A. We've got 3 fantastic technical talks. I'll introduce each one as we go along. And I think that's all from me, apart from one really good bit of news, actually. For those that don't know, we finally secured the CREST 2 funding. Now, I know we said we had it some time ago, but it's now been officially signed off. So you certainly have us for another 3 years, but hopefully we'll be well on our way to growing it into a really innovative centre then. And hopefully we won't be relying on ERDF funding. So that's a really good piece of news. And I know Kat, Vicky, Arta, and I believe Jane, who may be on the call, who are in the team, may also want to say something later about that. Anyway, I shall stop talking and I shall introduce Pete. So Pete is from the Shropshire Wildlife Trust and has been working in conservation, nature conservation for over 27 years. Pete's been a social worker, he's been an outdoor pursuits instructor, a centre manager for Coppice Crafts Centre for Adults, I believe. He's been in Shropshire since 2003, so a good 17 years here now. And within that time, he's been a tree surgeon and a hedge layer and a professional bodger. I love that, actually. Very Heath Robinson, love that. But for the last 14 years, Pete's been based with the Wildlife Trust in Shrewsbury. He manages all sizes of projects. He's currently the senior manager leading river and catchment management programs with various partners all around Shropshire. And as Kat said, Pete's going to talk about aquatic ecology tonight. So over to you, Pete.
Thank you very much, Juliana. I'm just going to do this and this and a little bit of that and do this. A bit of luck, that'll work quite nicely. Bingo. Okay. Really nice that the Shrewsbury Water Forum has basically the new communication technology, and hopefully my very general introduction to aquatic ecology will pave the way for our 2 genuine experts who will drill much deeper into the world of aquatic invertebrates and aquatic invasive species. So aquatic ecology, I say very general. What I'm going to do is just set out some of the concepts around ecology, some of and a couple of case examples of how that kind of works and helps us understand the natural world in a better and more scientific way. So let's see whether I can get that to progress. Here we go. So ecology is purely a study of relationships. And relationships between the biotic, the living parts of the earth, and the abiotic components of the earth. So that's your rocks and stones, your geology, and obviously the living creatures, whether that's your tiniest piece of bacterial scum floating around, or up to your elephants or giant sequoia trees. It's a whole lot in relation. And it's quite interesting just to check the etymology of these types of words. And oikos is the Greek word for home or place of where you live, and logi is study. So we're studying where we live. And as we start to peel away this general, so we've got our topic, what are we going to do with it? And we're going to start to map out the flows through that. We know it's a dynamic system. We know it changes all the time. And the drivers around it, rather like the sun and magma drives the world, is energy, nutrients, water, constantly shifting, the changing form of the time. And we're trying to work how those different things and different states and conditions are interacting and creating results. And those results are temporary because it changes. Living things die, inanimate objects get degraded and worn out, and they're transferred all over the place, and they shift and move. So that's what we're looking at, that constant flux of things. And what we do, we're inclined, rather like Linnaeus back in a couple of hundred years ago, is we want to understand that, and the first thing to do is to start to break it down into groups or a typology. And the major building block of an ecological understanding is an ecosystem. We can either take it at a global level, so the whole Earth as an ecosystem, or more usually, we look at something that's got a relatively defined character. So whether it's a mountain or a montane environment, or the coastal, the littoral area, or a grassland, it's got a particular characteristic, and that ecosystem will interact in a relatively easy way for us to start to understand. But these things are artificial because everything is connected. And what's quite useful, I haven't labelled it here as such today, is a term called the ecotone. And the ecotone is the gradient or place between two ecosystems. So between a grassland and a woodland will be an ecotone. Now sometimes they're quite large and the gradient between one and the other is very elongated, or sometimes it's quite abrupt. And it's useful to understand that it grades, but they're all connected in a profound way. So aquatic ecology is quite simply is looking at water-dependent ecosystems, and that's whether that's oceans and seas, which are clearly got paleophytic, salt-laden impacts. The chemistry is quite different, or down to your freshwater pond. Equally variable, infinitely variable, I'd say. But generally, aquatic ecology is about those either still water bodies or running water, or those oceanic bodies of water that are moving around. And clearly you've got ecotones between. The estuary is an ecotone between freshwater and saltwater. So as I've indicated, I quite like ecology. I quite like landscapes as well. I'm very much a generalist because the factors that are in play, there's lots of them, and you can approach it from lots of different angles. So this is a web map of the rivers of Shropshire. That represents about 3,800 miles of watercourse. Some are quite significant. This is the running water in the county. Those in the north are heavily influenced by the sandstone content in the geology. In the south, it's something else at play. Up where I am, up in the northwest corner, it's limestone, which may or may not have an impact because lots of the rivers are running in valleys and they've got glacial fills. So I could go on particularly about geology. Water quality, and I think later this evening we're going to have a much closer detailed look at the impacts of polluted water. But we need to bear in mind that water is impacted by atmospheric deposition, it's rainfall. Is that rain, which is generally mildly acidic, is it carrying particulate matter in it? Have we got a lot of it? So we've got water quantity, is another issue to take into account. And currently we're in July. We're being replenished today. I spent a nice day getting quite wet out and about down by the River Severn. But without quantities of water, sometimes that impacts on our ecosystems and creates an effect, and the relationships change. On a larger scale, we know we're in the middle of quite a conscious climate emergency. Our climate is those major trends around our small-scale weather systems, and that will change how our water-dependent habitats and ecosystems function and behave. What's happening to that dynamic range of changes, and that might dictate how we intervene in them, because we are a biotic component of ecosystems. It's not purely something that's other than ourselves. We are part of that. It's rather like an anthropologist. You have to take account when they arrive in that pristine environment, starts to to look at the culture, they've already made an impact rather like us. Finally, on this particular section, we're looking at biogeography. We know, because we're a globe, slightly squashed sort of globe, that we have different climates in different parts of the globe, and we end up with different ecologies having an impact end up with biogeography. So again, it's looking at massive scale. We sit on the kind of edge of the Atlantic, so we have Atlantic impacts. So a lot of our woodland on the west of the country starts to reflect that impact, oceanic impact. Whereas down in the southeast of the UK, we start to have a continental effect, and that will influence the number of species, the range of species. So a couple of concepts to track in here. Feedback loops. We've all encountered feedback loops. We maintain a lot of our own personal relationships through feedback loops. Stop doing that. You snore, all that sort of stuff. They're important in understanding ecosystems, because changes in one part of the ecosystem will create an impact, and that will feed back into the original action. We have competition we need to take account of, and there are finite resources, and sometimes we have to take account of those resources, because say an overheating of a water body suddenly become abundant. They actually attract a spurt and a thriving overpopulation of a certain species, and then that might cause a collapse once that competition has been resolved, or the resources have been taken away or consumed. So there's this constant interplay, and I think the competition resources, in terms of evolutionary change, has ended up in terms of developing what we call niche occupation. So each species has a certain set of preferences and needs in order to survive, breed, reproduce, and continue into the next generation. And they tend to adapt. And we can see, particularly in the invertebrate community, those adaptations in order to fit and feel their way into a niche are sometimes incredibly specialized. So the lost gall, and the lost goes into oak apples, or into marble galls, drills a hole, lays a larvae in. Now that is really specialized niche. No other creature behaves in the same way. So they found somewhere within the ecosystem that they can actually complete their life cycle. And I think we can look at the negative impact of the tidying and uniformity process, the uniforming process of our landscape, is the number of niches decline, the tidier and neater our landscape is. And that means that a range of species that can occupy have got less niches, and they're confined, and they tend to see declines. So that kind of understand that relationship between niche, occupation, competition. And finally, on this particular section, we've got carrying capacity. It's an important concept to get your head around. Is our pond, how much can it actually support? If we put in some large fish into our wildlife pond, and we put in too many, we tend to have an impact on that population, that top of the food chain predator will have an impact. It'll have an impact later down the food chain in terms of other species attempting to survive. So carrying capacity is important to understand, particularly when we're making decisions about manipulating ecosystems, and standing back from them and assessing what's happening to the energy flows. What is that capacity? What's the potential there on that side? A bit of a delay. Try that one. Oh, there we go. So, it's very much an introduction. We have some very complex cycles around energy. Energy is collected and used by plants to photosynthesize, produce oxygen and energy. Those producers, those primary producers, or autotrophs, are given them a sort of clumsy name, a sort of technical name. The primary users that raw energy from the sun on a daily diurnal basis, and they convert it into energy, into growth. They combine with nutrients, including the 13 micronutrients which are essential to life, to grow. And then the consumers, that secondary layer, secondary trophic layer, level which refers to where the sort of consumption of energy is done, will eat those primary producers to produce their own energy, or to gather their energy. So they have a different relationship because they can't produce their own. So it's important to start to understand those flows from those primal sources in terms of the sun, and how that feeds through, generates plant life, producers of plants, blah, 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 and consumers. Obviously, started to touch on the carbon cycle. We could have a separate session on the carbon cycle and how that works. But there's obviously clearly a cycling of those energies and those nutrients up and down and around and through, and some are stored for a long time, some are released inappropriately or rapidly, so the cycle starts to fall out of sync, and you get deficiencies or overloading in other parts. Another little concept, or is a large concept to add into this kind of just laying out of what aquatic ecology has to start to think about, is biomass. And it's important to think about the total values, or the value in an ecosystem of the living and dead organic matter, because the dead organic matter still has a role to play in the cycling of nutrients, in the productivity of the ecosystem and how it works. Sometimes you'll get a gain because your production rate is relatively high, and other times you'll end up with a loss. And it's important to have this concept when you're trying to understand why populations, the location of a target species are starting to decline, because the biomass that they're dependent on is also in decline. There's not sufficient being produced. And also it's important to understand that if you've got production rate of biomass, of living biomass, that's you end up with algal blooms, oxygen depletion, because you've got too much. You've got an acceleration of biomass. And certainly it's interesting when you looking at estuarine ecosystems, the biomass there is colossal, absolutely huge amount of biomass locked to estuary. It's really quite difficult to offer over because they're just muddy wastes, but they're supporting this incredible level of biomass of the living and the dead in perpetual cycles of biomass. So again, back to that sort of Linnaean sort of drive to start to break the ecosystem down and understand those relationships. We look at the consumers, and one way to sift those layers, those trophic layers out, is to look at carnivores, those that eat others, herbivores that predate directly onto your primary producers, omnivores that can swap in between the both, and then detrivores, so rather they're actually feeding on the dead organic matter that's part of that particular ecosystem. I've just given a couple of examples there of the heron, very much a carnivore, eats small fish. Fish are browsers, they graze on biofilm, that's that slimy stuff that's on plants in aquatic habitats. Some fish are obviously predators, who feed on fish that are omnivores. So you start to build up these relationships. You can see that the one without the other may actually be a problem. In relation to fisheries management, it's really quite important to have a healthy population of predatory pike, as I'm talking about a silver fishery, because their interaction with the other fish within that actually creates a much healthier community of piscine sort of occupants of that canal or river or still water lake. So breaking it down again. We can also look at consumers in a different way, and we can look at how they eat. Are they grazers or scrapers, or do they shred things up? Do they, rather like the freshwater mussel, actually collect and filter and gather up small particles? They're not abrading the environment, they're actually got a cleaning function. With the diving beetle on the bottom right, that's a significant predator, very aggressive, will bite you. And it forms that part of that chain, that interaction. So we have your predator who actually feeds in a particular way. And back to our pike example, pike are ambush predators. They lurk, and therefore they need foliage. They need a food chain that includes other smaller fish in them. So it's a full how do these species within the ecosystem actually eat? So again, very much looking at these big concepts without getting too much into the detail. It's quite easy in ecology to get lost in the detail. So we had a food chain, fantastic, that's kind of just moving up the trophic levels, and taking that energy from those primary producers into tertiary producers into secondary consumers. So that's an interesting progression. Clearly at the top you've got an accumulation of stuff happening, because lots more species are being brought up and their essential nutrients stuff are being accumulated in the top predators in the food chain, in the food web. But it's again looking at expanding that, make it a bit more complex, looking at the food chain, is understand those interdependencies of different species on each other. And the little diagram on the right starts to indicate how some of those species are caught into feedback loops, that one affects another without as a problem, because it needs that constituents to make it kind of successful. And others, like the heron, sits at the top there and actually picking off those things. So food chains are a useful concept to get into. And I know certainly many of you this evening will already be quite familiar with that concept, and many of the others that I'm kind of just reviewing this evening. Micro-organisms, they're water dependent. They form very, very small plants. We've got an image of some diatoms there, some algae. Some are motile, they actually move around. The blue-green cyana are a bacteria, that's the sort of bottom right. They're very, very small. And aquatic ecology can lead you from microorganisms, the small, up to the very large. And it's beginning to think, well, actually, what are those relations? Some of those are very dependent on nutrient functions in terms of the amount of nutrients that are available, and they explain pollution within the aquatic environment. But again, we're operating at very, very small scales. Moving up the sort of trophic level, or further up in terms of the autotrophs, we have woody plants, the trees, which tend to be marginal to aquatic environments, because generally the woody shrubs need their roots into terra firma. We have a whole range of plants that are emergent, so they can sit on the edge, the marginal emergence. The emergence also dependent on water that isn't particularly too deep. Floating plants, they're dependent on being able to pick up sunlight from above, so they can't be too deep. So they can most of the plant could be on the surface. They tend not to, they don't have to root particularly, they can actually move around. So duckweed's a really good example of that. A very small plant, the lemna, has a tiny little rootlet, hangs down, but they'll float on the surface. And you'll find them on rivers as well as ponds. But they're producing by photosynthesis oxygen, so they're important. And the Canadian pondweed on the bottom right there is an oxygenator. It's producing oxygen into the water. The bog bean on the bottom left is an emergent, so relatively shallow water. And some plants are very specialised. They're occupying peripheral or ephemeral pools that actually are there very, very briefly indeed. So I'm just going to make sure I stay on time. I'm aware that we've got only a certain amount of time here. We've got invertebrates, huge group. Remember I was talking about niches, how their life cycle actually fits into the ecosystem to the aquatic one. And obviously the aquatic ecology, you're focusing on aquatic species. Unfortunately, some of them are quite easy to and straightforward. So there's only one aquatic spider, the water spider. But when you get into flatworms and flukes, you're suddenly into quite a complex area. I quite like flatworms. I think they're good fun. Lovely little sort of kitten-like creatures. And clearly we have some large things like the crustaceans, of which we have a crayfish example. Moving up the trophic scale, we have some sort of top of the chain type creatures. So we have our fish, large range of fish. Some fish are androgynous. They actually don't mind being in fresh water and salt. So our shad, salmon, sea trout, brown trout, same species. They're just tolerant of both environments. And obviously amphibians, have majorly adapted. And I think that's probably the point of this slide, is some of our mammals and birds, though not as fully adapted as fish to aquatic environments, have adapted up to a point, and the water shrew is affected because it's adapted to water environment, and it performs very well underwater. It can swim in let's see if we can advance the slide. There we go. So having set the scene, we have aquatic ecosystems. We have, in gross terms, those impacts that are negative or positive. Is you have to look at water quality, quantity. We've got too much, too little. Is the pond dried up? As the river dried up? What's its resilience? Bioaccumulation, I know we're going to come back to in one of the later presentations. Biomagnification, we've got an impact of a pollutant where the impact is just growing because of other factors within that ecosystem, whether that's temperature or the quality of the rainwater at that particular time. Atmospheric pollution, as we know, can pick up particulate matter. Acid rain was a significant issue that raised and made it very clear that habitats. The slide just illustrates endocrine disruption. So we have a hormone system. Most species have one of some sort, and chemicals that cause endocrine disruption can actually cause a whole range of mutations, hybridizations, illnesses and conditions, and having those in an aquatic environment. And water is such a massively effective diffuser and carrier of pollution. So physical damage to our ecosystem, and then these massive biogeographical global climate change factors have to be taken into account, where species distributions start to change, the availability of water, temperature ranges for breeding. Won't dwell too long on this, but I'm sure Martin will come back and with his presentation look at negative cascade interactions, where a species is introduced into an ecosystem, and we get a whole range of follow-on reactions and interactions caused by that single change. So just a couple of examples of these kind of feedback loops. We clearly have invasives that are aquatic. They're quite mobile. They're not just brought in by human impacts in terms of us carrying them on fishing kit, machinery, or boats or shipping or whatever. They can actually travel quite effectively themselves. This particular species arrived in ships in ballast water, blocks up drains, so it has an impact on the human part of the system. But notably, it will actually just browse away and collect all of the plankton in the system, and you get an imbalance in terms of what's available to juvenile fish in the same system. So clearly they're out of balance, they're in the wrong place. and they'll create a problem or an issue. So another little, just an example of a niche cycle. They are very dependent on the abiotic component. They like clean gravel beds. They need those gravel beds to be loose in the right place. Water temperature and quality needs to be within certain ranges, and if those aren't present, along with abundant food source, then the brown trout, because it has this large complex life cycle, will have a challenge to survive and complete its breeding cycle. So I think that's just a good example of the kind of complexities of the ecosystem, how it affects one of the significant keystone species within that system. Just much debate is about these three characters here. It's got the water vole, American mink, and otter. The otter has seen a phenomenal recovery because pollutants and were removed from its environment that it was in. But subsequently, we also back in the early 20th century, we have the introduction of the American mink. Mink are smaller than otters, but they're very good in terms of general purpose. They're quite adaptable, part of the same mustardid family. But on our left-hand side, we have the water vole, less well adapted, quite a fragile, has a burrow system, so its habitat demands are quite great in terms of its niche. Requires a range of foodstuffs within its chosen habitat, and it has relatively, certainly in terms now, relatively isolated populations. So it's dependent on those ecosystem connectivity across a landscape. And that does form this complex picture between the otter that occupies a territory, the mink that can interplay within that territory, and if habitat is degraded and poor, the mink will predate on the water vole. But the otter will push the mink out slightly. Still not resolved, that one. I think that's the appeal of ecology, is working that one. We have a little bit of a niche adaptation example again. They're relatively widespread kingfisher. They're adapted to an aquatic environment, so they have a transparent fluid eyelid, effective fish, so they're a predator in terms of their trophic presence. They're not top of the food chain, but they're up there. They are territorial, so they need good habitat in sufficient quantity. And they also, in order to successfully reproduce, they require riverbanks, actively eroding riverbanks, not too active, in order to build their burrows. So it's an interplay of the abiotic environment, of having appropriate sandy riverbanks that they can burrow into, sufficient prey at the right size, because they're obviously determined. They're a beautiful bird, but they're relatively small, in order to make those balances within their chosen adapted ecosystem, and they occupy that quite clear niche. So they don't eat fish as big as a heron, for instance. That's very much a crack whip through introduction to aquatic ecology, and I believe I'm going to hand over and stop sharing.
Our next speaker.
And
thank you very much, Pete. Juliana, do you want to introduce our next speaker?
Yeah, I will do. Yeah, thanks again, Pete. That's really interesting. Okay, so we've now got Martin Fenn from the Environment Agency. Martin's worked at the EA as a freshwater ecologist for over 18 years. He's also based at the Shrewsbury office, but he covers the whole of the West Midlands area, so massive area to cover. Martin leads on the fish monitoring programme, biosecurity and invasive non-native species. He also sits on the national EA group entitled Invasive Species, this is a mouthful, Invasive Species Biosecurity Network. And as part of this network, he chairs the Knowledge and Innovation subgroup. That's much easier. Martin's going to talk to us now about aquatic invasive non-native species. We should look forward to it. Over to you, Martin. Thank you.
Thank you very much. I'll just hopefully start sharing screen and we'll be up and running. So hopefully everybody can see my screen okay now. So I'm going to quickly talk through aquatic invasive non-native species. Through the talk, I might refer to them as INS, just to speed things up a little bit. You might have also heard the term invasive alien species as well. And that's one that's been developed through the European Union and the directive that they brought through in 2015. So, but we'll try and keep it through with INS for now. So hopefully we'll start moving through the slides. For some reason it's not scrolling on. Ah, there we go. Book summary, we're just going to go through really the basics on what invasive species are. Why are invasive species a problem? And then we'll go through some examples of aquatic invasive species, focusing on some of the more common ones that we're likely to come across. And then just finish it off really with what we can do about them. Is there anything we can do? So, invasive non-native species. We have classed these in various ways, but the most easy way to put it is that a non-native species is an organism which has been introduced outside of its native range, and this is usually through the actions of humans. So, for example, for the UK, after the last ice age, 10,000 years ago, we had species that naturally repopulated the area. But then over time, humans moved into the area, and then they started moving species across as well. And it's these human interactions that cause them non-native species. Now, only a small proportion of these non-native species become to a point where they're actually invasive, and they start having a negative impact on the environment, the economy, or even the social impact with some of them, which we'll go through later on. Just to say, a rough figure, probably about a quarter of invasive non-native species within the UK live within the freshwater environment. So that's an important part of the system. I am going to mainly stick within the freshwater component and not go into the marine, because that is a whole different ball game and a lot more complicated. So where do INS come from? I suppose this could also be phrased a bit more of how do they get here? So we have several major sort of routes that they get into the country, especially historically, a lot of plants and animals have been brought in for the ornamental garden trade. Unfortunately, what happens then is that they often escape from the gardens or are actually physically removed from the gardens because they're an issue within their garden environment, growing too much, and then dumped into the wild. We then have exotic pets that escape or are released into the wild. These tend to be not so much of a large issue because of usually they're generally just one-offs, and so therefore they very rarely start a breeding population, and so can be controlled quite easily. We have commercial species that escape or are allowed to establish in the wild. For example, the American mink that Pete mentioned earlier. And then we have these that we call class as hitchhikers. So these that are adapted to be able to clasp onto something, whether it be through transport or they're clinging onto clothing or equipment, and they're able to actually move with human movements into new areas. So that is how they get here, but why are they a problem? What tends to happen is when they move to a new environment, the invasive species hasn't got any of its controls that it would have in its native range. So all of its enemies that are there that usually help control the population size are not there anymore. And so that's when they start to reproduce rapidly and take over. They often have an unfair advantage in their new environment and can quite easily outcompete native species. Unfortunately, the freshwater system is exceptionally vulnerable to invasion. Water acts as a really good dispersal corridor for invasive species. And so once they get established, they can quite quickly spread, especially within flowing water systems. And invasive species are recognised as one of the biggest threats to biodiversity globally, and so is therefore noted worldwide that this is an issue that we need to tackle. We'll focus a little bit more on the impacts of invasive species, and a lot of these are ones that are really important to the Environment Agency, but are important to everybody else as well, really. Quite often in these examples, there's a lot of invasive plant species especially that can cause a flood risk, whether that's through adding more bulk within the water or clogging up important infrastructure that's needed for the water to move out through the systems. It can also increase the cost of building works and cause issues which mean maintenance of important structures. They've reduced the ecological quality through, whether that be through disease, competition, predation. Obviously, there's usually quite large interactions with these, as stated by Pete earlier. So all these things work together, and it sometimes is very difficult to pick out what they're actually their main way is that they're impacting on the ecology, but sometimes it becomes very obvious. They also degrade recreational activities, especially things such as angling, canoeing, for example, making it very difficult in some rivers to actually partake in these activities. Reducing navigation as well, especially in the canal network, where sometimes certain species like floating pennywort can clog up the canals. They will erode riverbanks and increase siltation, which has an impact on several species. For example, the brown trout, where it requires the clean gravel beds to be able to reproduce. And as we see it, invasive species are potentially one of the worst type of incidents that we have to deal with. So, for example, if we get a pollution of a slurry going through, that goes through the system, kills a proportion of the ecology within that riverbed, but it passes through, it dilutes as it goes downstream, and the river is able to recover. But in invasive species, when that invades an environment, if nothing is done about it, it just expands and causes further issues there and further upstream and downstream. So in that respect, it makes them a lot worse. So I thought now I'd just go through some of the more common invasive species that you may have come across. On all these, I'll try to explain a little bit about the species itself, where it's come from originally, and some of the impacts that it might have on the ecology. Also, just to state that the map that you see in the right-hand corner there, that's showing the most recent distribution that we have for that species in common. So for the Himalayan balsam, that was introduced way back in 1839, and there's records to show that that was actually moved first of all into Kew Gardens. It was brought in as an ornamental plant species. Its distribution, as you can see, is widespread throughout the UK, not quite so densely populated in the hills of Scotland, but otherwise it is fairly widespread. It is one of the largest annual plants that we have in the UK and can grow up to 2 meters in 1 year, It forms very dense stands and outcompetes native species. As you can see in the photograph there, it's a full monospecific stand, as we say. There's not really any room for anything else to grow. The pungent flowers attract pollinators, and that attracts pollinators away from our native species, and so therefore has a knock-on impact there. Also, these plants have a shallow root system, and because of their annual, they die back every year. And as they die back, that leaves riverbeds, riverbanks exposed, causing siltation as the silt from the bankside goes into the rivers. And they have a very impressive dispersal mechanism. They have explosive seed capsules, so they're like really tightly coiled springs. And so as they dry, the seeds pop and they can travel up to 7 meters in distance. Add to that then that the seeds can float within the river systems, means that they've got an excellent way of being able to disperse down river corridors. And this is why when we try to manage Himalayan balsam, we always try to start at the top of catchments and then work our way down. Second species we're going to look at is floating pennywort. And this is more of a recent introduction to the UK, hence really why the distribution is mainly, well, it's a lot less, as you can see, and with most of the clusters down in the southeast of the country. And this invader comes from the Americas, and it was brought in as an ornamental plant for ponds and aquarium, as an oxygenating plant. Unfortunately, though, it forms these really dense floating mats in still or slow-flowing waters, and the speed that this plant grows is ridiculous. It's been measured in optimal conditions to grow up to 20 centimetres a day, and so therefore it very quickly chokes up riverbeds. This involves outcompeting native plant species, but it also blocks light getting to the riverbed, which is obviously an important part of the system. In winter, this dies back and can increase nutrient loads to the water. But it also disrupts navigation, fishing, and can also lead to flooding. Also, as you can see in the picture in the bottom left, it can be a hazard. Animals and even humans can sometimes not realise that that is actually water below there, and they get trapped and can sometimes even drown within the situation. The main reproductive strategy for floating pennywort is through vegetation and vegetative reproduction. So small fragments are able to reroot and grow on. And so you have to be very careful when trying to eradicate or take this species out, that you don't leave any parts in the watercourse, or that you accidentally knock off some of the actual floating pennywort, which then could float on downstream. Next species that we're looking at is the signal crayfish, a crustacean brought in in the 1970s, and was brought in as farmed for food, really. Even though the main market for this is in Scandinavia, it was felt that bringing this species in from its native North America would be financially viable. Unfortunately, it's spread, as you can see, through much of England and into Wales and parts of Scotland as well now. The problem is this is a much larger and more aggressive species of crayfish than our small native white-clawed crayfish, and it easily outcompetes it and even predates on it. But the largest problem with the signal crayfish is that it's a carrier of a fungal disease called crayfish plague, which our native crayfish have no tolerance to. And so therefore the fungal disease can quite quickly wipe out our native crayfish populations, and that has seen a real plummet over the last 20 to 30 years. Also, aquatic ecosystems see a lowering of community diversity and species richness after invasion of signal crayfish, as they are large predators that will eat a large variety of food. But also, especially in riverbanks that have got soft riverbanks, they form burrows which destabilize the banks and cause bank erosion, adding more siltation into the rivers. The signal crayfish are able to disperse up and downstream through crawling and even can navigate certain barriers. They're also able to cover reasonable distances over land. They've been measured to cover over several hundred meters in one evening. This is one of the ways how they were able to escape from fish farms in the first place. We'll look at a slightly smaller individual this time, and this one is the demon shrimp. At present, we're unsure how this species was introduced to the UK, but we think it was through the mechanism of hitchhiking, which is it's very good dispersal mechanism for this species. It was first recorded in the UK in the River Severn near Worcester in 2012. However, it soon became apparent that it was throughout the English canal network, and from there it has spread throughout much of England, unfortunately. It is native to the Ponto-Caspian region of Eastern Europe, so that's sort of like the Black Sea area, and has spread through much of Western Europe, mainly through linking of river systems through canal networks. The demon shrimp is the slightly less aggressive relative of the killer shrimp, which was also found in this country back in 2010. However, the killer shrimp has mainly been able to be stopped in its spread because it's been found more in enclosed water bodies, and so through strict biosecurity we have made sure that that hasn't spread to the same extent as the demon shrimp. This species is still mainly predatory and will eat a wide variety of macroinvertebrates, including our own freshwater shrimp. And unfortunately, that causes real issues within the ecosystem and the food web. Our native shrimps are leaf shredders, and so therefore removing them from the system will have knock-on impacts within the ecosystem. The demon shrimp itself has a very high reproductive rate, and its ability to cling to surfaces such as boats and waders have made it a very efficient invader of water bodies. This is probably one you're all aware of, Japanese knotweed. This again was a plant that was introduced in the 1800s as an ornamental plant, which has escaped out into the wild. And as you can see on the little map, it is now widely distributed throughout the United Kingdom. Again, it forms really dense stands, outcompeting native species, and when it dies back in the winter, leaves riverbanks exposed to erosion. Another real problem with Japanese knotweed is that it can cause structural damage to buildings and infrastructure. In Japan, where it originates from, this species is found on the sides of volcanoes and is one of those pioneer species that are able to grow up through the lava rock as it's solidified and be able to establish themselves there. So therefore they're able to quite easily grow through brick structures, through tarmac, and they can cause real damage, and now are a real issue for homeowners, and can sometimes mortgages can be stopped because of having Japanese knotweed in the property. The distribution or dispersal of Japanese knotweed is mainly vegetative, except for along river corridors where rhizomes, the root pieces, can be washed down during flood events. Also, those rhizomes can be moved within topsoil if it's been contaminated from a site where Japanese knotweed was originally. And we can quite often see Japanese knotweed being spread through that method. Giant hogweed. This was introduced again back in the 1800s. The Victorians seem to be, with their adventures throughout the world, they brought back a number of wild species. And this was brought back as a curiosity species, really. It's now widely distributed through the United Kingdom, but it's a native of the Caucasus Mountains. And so that's a sort of the mountain range between the Black Sea and the Caspian Sea. This species again can form quite dense stands, but they're very tall. This species over a couple of years will grow to 2 to 3 meters in height. And obviously with forming these dense mats, they will then outcompete native species. However, the main problem that we have with giant hogweed is the fact that the sap that is held within the plant, and if a human comes in contact with that sap through the hairs that you find on the majority of the plant, that sap is phytotoxic, and so therefore can cause blistering on the skin and rashes. And this can last for a number of years. And so frequent exposure to sun afterwards can still cause the skin to blister and rash. So this is a real health impact and something that we need to be very wary of if we find it out in the wild. Last species on the common species that we're looking at is the American skunk cabbage. First introduced as an ornamental plant in the 1900s, another native of North America that's been brought over and is mainly found in wet woodlands. It's widespread through the UK, but not generally common. So it's usually small pockets of them rather than the widespread nature of, say, Himalayan balsam or Japanese knotweed. It forms, again, dense monospecific stands, and research has shown that it has a significant impact on riverine woodland ecosystems. It's a long-lived perennial plant, and it mainly reproduces through seeds. However, also it can disperse through rhizomes. And yes, it does smell supposedly very similar to skunks, hence the name. Some of the other notable aquatic invasive species that we should be aware of in the area, we have Australian swamp stonecrop, sometimes known as New Zealand pygmyweed. Some people might know it by its Latin name, Crassula helmsii. It's another one of these invasive plants that is emergent but can also then grow up onto the sides, and it is a real pain to try to get rid of. It's very resistant even to drying out, so trying to eradicate this is a real issue. Then two of the other species, so I won't touch on too much because of Pete covered these in his presentation, but the zebra mussels have a cause real problems to the water industry, clogging up pipelines. And the mink, as we've seen earlier, not only does it impact our water vole populations, but also it has impact on our fish populations as well. What we do also have is species that we call alert species. So these are species that are relatively new to the UK, or maybe not even in the UK yet. And the idea is that if we can eradicate these species early on, it saves us a lot of cost in the long run. So we have creeping water primrose, which is an invader from South America and has been a real problem over on the continent in Europe. However, we only have a handful of sites within the United Kingdom, which we're hoping we are managing as the Environment Agency at the moment to eradicate these. They are quite long-lived and quite difficult to eradicate, so it's a slow process. But we've been managing these for 10 years now, and I think the amount of water primrose left in the UK is now down to hundreds of meters squared, while previously it was in the thousands of meters. So we have a real chance of eradicating this species before it gets a foothold. Another species is the fish, the topmouth gudgeon. We have had a couple of these crop up in pools around the West Midlands, and we have been able to eradicate these using a fish pesticide, so rotenone. And the idea is that we think that we will be able to eradicate this species, mainly because we've been able to find them in still waters fisheries rather than rivers. If we start finding them in rivers, that will make it very, very difficult. And then I've just mentioned the Asian hornet here, just very quickly. It's not an aquatic species, but it is a species that we're really concerned about. And we want to try to make sure that any new sightings of this species coming from Europe are found quickly, so we can then get rid of the hives and stop them reproducing. And at present, we've been very successful in this, and so we don't have a large population, unlike the Channel Islands, which have got a real problem with this now, as they've got a very active population. The real problem with Asian hornets, just to quickly mention, is that they feed mainly on honeybees, and so our bees are already in real trouble, so we don't want to add an extra vector there that could cause them issues. So what can we do about this? Management and eradication is the main thing really that we should be concentrating on. Some species are easier to manage than others. Japanese knotweed is one of those that's really difficult. Because of everything is stored in those rhizomes, you have to be able to kill it off and kill those rhizomes as well. So even with herbicides, those rhizomes can sometimes stay dormant for quite long periods and takes a long time to eradicate. Season can be important as well. For example, Himalayan balsam. There's no point trying to pull out Himalayan balsam when it's flowering and in seed, because you're just probably going to cause the problem even more. The ideal is to start pulling the Himalayan balsam. It's shallow rooted, so that's easily done, but do it before it starts flowering and setting seeds. And the topmouth gudgeon, we're having to do that over the winter months because of the eggs from the topmouth gudgeon can be quite resistant to the poison, but they only reproduce when the water temperature gets to a certain temperature, and so over winter they don't reproduce. Biocontrol, this is a really new important science that we're taking on, and CABI are the group that run thousands of trials on these before they're let out into the environment, and these are found to be really specific species which will only attack the invasive species that we're involved with. And so we have the Himalayan balsam rust fungus, we have an aphid that's attacking Japanese knotweed, and there's several others really important biocontrols which will hopefully weaken these species a lot. And just to give you an idea that Great Britain spends at least £1.7 billion a year on control of invasive species, and that was calculated back in 2010 by DEFRA. So actually that figure is probably even larger now. Important to know your enemy. Get to know the invasive species that you're dealing with. The GB Non-Native Species Secretariat is a really, really good website to go to, and I would heartily recommend anybody interested in invasive species having a look at that. There are also a number of good field guides. that are available. One that we use quite a lot is this field guide produced by Olaf Boy, Max Wade and Helen Roy, but there are plenty of others out there. Recording invasive species help with knowing the distribution, so we can help with the management, but also the biosecurity. With smartphone technology, this has improved drastically. So we have specific apps now, for example, the Asian Hornet app, where you're able to actually record any sightings. And the great thing about this is also it has ID guides. So it helps you be sure that what you're taking a photo of is the Asian hornet and not the European hornet. We also have things such as InsMapper, iRecord, iNaturalist. There's lots of systems out there for you to be able to record our invasive species and help us learn a little bit more about their distribution. Biosecurity, this is the really important step. So all those people that go out into the environment, the water environment is especially important, but even if you're going out walking your dogs, things like that, it's so easy to transport invasive species. So these are the 3 easy steps to make. So first of all, it's check your equipment and clothing for organisms, especially if you know that you've been in contact with an area that has invasive species, especially with water. Damp places are the really things. So if you go fishing, for example, and you've got a net and a kit bag, that will stay damp for a long time. Invasive species can stay and live in those conditions for quite a long time. Clean and wash equipment where possible, especially on site if at all possible. If not, when you get home, it's really important to try to clean off. Hot water is very good at killing off invasive species. Some disinfectants are also very useful. And drying, especially for freshwater species and animals especially, drying is a really important part. Because they live within water systems, actually drying them out thoroughly for 48 hours is a really effective means of killing them off. What I did want to do was just to finally really just a brief shout out to CINSI, which is the Shropshire Invasive Non-Native Species Initiative, which has been set up by the Shropshire Wildlife Trust. They've been able to get some funding to produce this. It's on their website and it's a really useful resource, and hopefully we'll be doing a lot more work within Shropshire with this group to improve our understanding of invasive species, but also our management. So any help you can provide for that would be much appreciated. Any questions?
Any questions?
That, Martin, it was really, really interesting, and now I know whenever I hear the word INS what it actually means, so thank you. Okay, I'd like to introduce Lucy, Dr. Lucy Crockford. Lucy's joined us from Harper University tonight. Lucy is a senior lecturer in soil and water management. She completed her PhD, all her education actually in Ireland, in catchment science at Trinity College in Dublin. And before that, she completed an MSc in engineering, environmental engineering at Queen's University, and then also an undergraduate degree at the National University of Ireland in Galway. Lucy she says she's a generalist, as in sort of an environmental science generalist, but equally she lectures in modules around ecology of aquatic ecosystems and pollution transfers to the aquatic environment, so incredibly relevant to this. Lucy's also been involved in lots of research projects. One of her latest projects has been around natural flood management, and in particular looking at groundwater fed streams and the accumulation of contaminants in the wider ecosystem, which I believe she's going to talk about now. So over to you, Lucy. Thank you.
Hi Juliana, thank you very much for the introduction. So yes, I'm Lucy and I'm a senior lecturer at Harper Adams University. And I'm here to talk to you a bit about environmental pollution and accumulation of these things in the environment. So, as may be obvious, I actually am from Ireland. So I grew up in a location which had a very large road that was going through this forest, and when it was being upgraded, it was expected to lose 9% of that of the forest. So we had some protesters, and as a consequence, they managed to reduce the amount of trees down to only 2% lost. So that really impacted me, and so from that really early age of about 12, I became really interested in the environment and have kind of spent all that time now, as was talked about just before. So what I want to talk about today then is talking about bioaccumulation. So it was mentioned briefly by Pete. I do have a case study to actually talk about then, and the findings from that case study. So I'm going to focus a bit on heavy metals. I still am very interested in nutrients. I love phosphorus, particularly. That was what my main focus was. But in the last while, I've been becoming more interested in heavy metal contamination. And these come from things like transport. We've got things like industrial processes where we have power generation, mining, agriculture, of course, is going to produce some heavy metals, particularly if we're using anything that was sewage sludge based into our system. And then of course we have rocks. So any sort of catchment is going to have heavy metals come out of the rocks due to the rainfall on that location. And those metals include things like lead and cadmium, mercury, aluminium, lots of various things that will come out. So how do we measure that? Well, I think most of us will be aware that we can take a sample and we can then measure it for the concentration of the contaminant. The problem with that though is that that sample was just taken at that time. And it could be that there's something happening in the catchment on a Tuesday and you took that sample on a Monday. So the water's had 6 days to get cleaner, and that sample then isn't going to be really relevant to explain a problem that might be happening in that environment. And so what we can do then is look at the biology of a system. And one thing we can do, particularly for air pollution, is to have a look at the lichens that are in the location. A lichen is a mixture of an algae and a fungus, and they're really intolerant to really poor air quality. And so depending on which lichens are present, you can get an indication of the type of air pollution that is happening there. We can also think about the presence and absence of other invertebrates, such as, for example, sandworms, also very intolerant to heavy metals in particular. And so we'll see these maybe in on the seashore and things as well. Another way we can look at trying to measure pollution is the absence or presence of visible indicators. So for example, in sycamore, if you have tar spot, this actually indicates good air quality, because essentially that fungus that causes a tar spot doesn't like sulfur dioxide. And you'll see in this table that we have a number of sites, and the sites that are in red are a really high level of sulfur dioxide, and the ones in green are really low. And if you go then to see if there's any presence or absence of the tar spot, then we can see that these match up. So the ones that are in red had really high sulfur dioxide, but no tar spot. And then we've got the ones in green, which had particularly low levels and have the presence of the tar spot. And you would expect then, as things become cleaner, we'll see a change in the ecosystem, that we start to see tar spot to come back into sycamore, particularly in our urban areas where it had pretty much disappeared. There was a project done last year by Opal. So it was a citizen science project, and they found actually the tar spot hadn't actually returned to sycamore. And in fact, the fungus was quite intolerant of nitrogen dioxide. And also because we clean our streets, it's really hard for the fungus to like move particularly easily. So when we're thinking about pollution, we have to be really clear about the pathways that the pollutants take, the overall ecology that indicates things. And that's why we can also look at ecological methods. So polluted streams, they result in the abandonment of traditional spawning areas and ultimately in the loss of salmon populations. Species sensitivity to pollution is quite variable, and so we can, map these things out and see where things have actually changed. So for example, in the Thames, which flows through London, this is probably the first major river to lose its stock. The last salmon was over a century ago, which is in 1833. And I meant to check if there's been any recently, because they have done great work on cleaning the Thames. But we've also seen a loss of salmon in South Wales, in the Taff, as a result of coal mining and metallurgical industries, and also sewage from the expanding population. However, one way that we can look at the quality of rivers, and something that I say a lot of people on this call have done, is we look at freshwater invertebrates. So these can indicate the water quality based on the levels of oxygen in the water. Some of our species are more tolerant to low oxygen conditions. So when we have lots of phosphorus and nitrogen and lots of algal blooms, we see a change in the oxygen dynamics in that river. The problem is, though, as I intimated, that sometimes we need to have a longer term measure that will indicate what's happening. And for heavy metals, it's quite difficult to be able to do that. However, we could look to see if the invertebrates are accumulating those metals in their tissues. So we need to then think about, as I said, how these contaminants move, where they go. And there's various factors that will have an impact on that fate. So for example, localization. At the source of the contamination, you're going to find obviously much higher concentrations of things. And so you can pinpoint where things are happening in a catchment based on that. And basically, if you do find a high source, often we'll try to just dilute that source so that it has a lower impact on the environment. Also interested in the persistence of the contaminant. There could be a fairly long residence time in soils and sediments. And sediments, of course, are really important for rivers and lakes. We're also interested in the bioavailability of that contaminant. Can it be taken up by the particular invertebrate? And I'll talk about that a bit later on too. Because we're interested in bioconcentration, bioaccumulation, and biomagnification, which I'll talk about in a bit more detail. So, what do I mean by our bioconcentration? Well, this is essentially how much of the contaminant is in the organism, in that river, in comparison to how much is in the water around that particular invertebrate, whatever it is that you're actually measuring. And so we could look at possibly the concentration in mayflies or in caddisflies. And you'll find actually, if you do look at any of the literature, that they tend to use bioconcentration interchangeably with the word for bioaccumulation. And I suppose the main difference is that for the bioconcentration, it's about aqueous ambient environment, whereas our bioaccumulation is about any of the ambient environments. And so we'll see bioaccumulation happening in things that we grow in the soil. So for example, we have some carrots here, which are going to be taking on some of the metals that are in that soil. And that's why we're so careful about where we grow our plants that we need to then consume, because those plants are going to take up what is in that soil. And we're really careful about what types of plants we grow in agriculture for that reason. Biomagnification then is literally where we have an increase in the concentration of that chemical or pollutant as you move through the food chain. So this is a food chain here. You'll see it starts with the mayflies, which are going to be that second order as talked about by Pete earlier on. So they're going to be eating litter, they're going to be eating plants, you know, to then also be consumed by, in this particular picture, a bluegill, which is a US fish. This is a picture from a website which is in the US, but it's basically a bream type of fish, so kind of smaller than what you find for a bass fish, which you find in estuarine areas. And then of course we have the human. And as you go through the food chain, you see the increase in concentration of the pollutant until it becomes a really big issue for the human that is eating all of this highly contaminated food. And we're what we call an apex predator. So with this kind of in mind, then I'm going to talk to you about this case study that we were working on. So it was an MSc project by Eric Dexheimer. He was doing his MSc in entomology here at Harper, which is all about the study of insects. And over the last few years, I've been working with Farm the Flow, which is the brainchild of Russell Rowley, who is the CEO of the Seven Gorge Countryside Trust, and also John Bellis, who previously was the flood engineer for Telford and Wrekin. And we saw him speak back in February time, I think it was. I was invited to join the group in its early stages to contribute my expertise in the management of water and to see if there's any areas that could be researched from a university point of view. So you can see the catchment there. Some of you will be familiar with it. We've got Colebrookdale, and we're kind of interested in the area going up this way. I'll show you another picture in a second. Here we go. So this is the upper part of the catchment, and this is what we call the Lydebrook. And in fact, it's two arms. Unfortunately, the second arm doesn't come out in the Catchment Data Explorer. But the two arms anyway, the one on the right is called the Lightmoor. It's much more urban. You can see that with all the houses and things in Dawley. It has some flooding issues that have been kind of worked on. And then on the left-hand side, we have what we call the Loam Hole, which is a much more agricultural location and is the catchment that I do most of the work in. So it's a triple SI for a cranefly larva, and also importantly, it had some dumping of pulverized fly ash in the 1950s. So there's quite a lot happening in the catchment, and because of the background geology and also with the dumping, there's a possibility of there being some heavy metal contamination occurring. It was definitely worthwhile having a look anyway. The catchment, like many farmed areas in the UK, is experiencing massive sediment movement. This is a picture of what we call cattle poaching, so not stealing cattle, but it's actually them standing at a gate and causing the soil to become compacted, and then it moves and things. And so we see this massive move of the sediment due to topography, and then also the previous dumping has loosened that cohesiveness of the soil. And so during periods of intense rainfall, levels in the water will rise quickly, can carry large trees as well as large quantities of soil. So just a few pictures there. There's the sediment trap where they have to clean it out every second year. So predominantly interested in kind of flooding in this catchment, but we thought it'd be worthwhile doing a project on the accumulation of contamination in the biological environment. So we had two proposed pollutant linkages. This is what we call when we're talking about the pathway of a contaminant through that food chain and also through the environment. In this case, we felt that by measuring the contamination in the freshwater invertebrates, we could get an idea of the extent of the movement of the waste from source to a receptor. So Eric and myself collected invertebrate samples from two locations in the catchment. So we chose a spot that was above where we thought that the actual dumping had happened, so up here. And then we also took some samples right where we were expecting there to have been some impact. The problem we had though was that the invertebrates that we found in this location would only be applicable for this catchment. And we didn't really have a reference point for what was high and what was low. So we did a road trip and we headed out to Wales in the hope that we'd find somewhere far away from industry that had a similar topography and also had a similar land use in the vicinity. So you can see there we've got lots of deciduous trees. It was quite steep, and we felt that it was a good kind of fit for trying to have something in a comparison. So at each site then we did our kick sampling. So some of you will have done this before. This is where you first of all do a search for a minute on all the stones. Then you stand in the river and you kick the sediment into the net, and you catch things in it, and then you also search the stones again for anything that you might have dislodged over that time. In addition to which invertebrates might be more abundant and therefore would give us enough biomass for our testing, because don't forget we want to be able to extract the metals out of the invertebrates, we needed to consider what we would be catching and the pathways that the metals may use in the bioaccumulation process. In these two examples we see on the left, we have an invertebrate with their whole life cycle in the water. So this could be a freshwater shrimp, and on the right we have a mayfly life cycle where there's a significant amount of time, 1 to 3 years, spent in the water as nymphs, and then they emerge as adults, usually in kind of late spring, early summer. Having a life cycle in the water for a number of years would be very useful. Unfortunately, shrimp only live for about 1 year, and they could do a few more. There has been 12 years mentioned, but mostly it's about a year. And unfortunately, we didn't have any filter feeders in our catch, which would have been the most appropriate type of invertebrate, as they accumulate lots of particulates by their feeding mechanism. So we're thinking about all these pathways. What do we do next? Well, we took all those little invertebrates that we caught, we mushed them up in some acid and some bleach, and we then put them through a fancy machine called an ICP-MS. And this was able to identify the metal concentration in this extraction from the invertebrates. And this was facilitated by the University of Wolverhampton by Professor Craig Williams, who was fantastic. What did we find there? Well, some of you will actually recognize these guys. At the top, we've got the mayflies. We've got 2 different species, well, 2 different families there. And we can see we've got 3 tails, which should tell most of you that that's a mayfly. At the bottom, we have our stoneflies with 2 tails, and then we have some chironomids, that's in the C box, and you also have the freshwater shrimp in the D box. So nothing particularly astounding. We were kind of expecting to see these sorts of invertebrates. So interestingly, the community structure was quite different between the 3 sites. So the 2 sites in Shropshire were quite similar because they had lots of shrimp, whereas in the Welsh catchment we actually found lots of worms. So really quite different catchments, and in the end we decided that we would digest Plecoptera and Ephemeroptera, so stoneflies and mayflies, the shrimp, and the caddisflies or Trichoptera. And these are species that are used quite frequently in science for determining the accumulation of metals. Those of you that have done a little bit of science may know about species richness and the Shannon index, so I just put those numbers in. You can see that they're quite similar. Our study site, which was where we expected there to be an impact, actually had a better Shannon index than the control site in Shropshire, which we thought was kind of interesting, but not largely different to the Wales site for kind of the quality of them. So what did we find? Well, we've got some graphs here. We can see we've got the 4 different types of invertebrates that were tested. On the y-axis, you can see that we've got the percentage of dry mass that contains this particular type of metal. So the first one being lead here. And we can see that there's quite a large amount of lead in the Plecoptera at the Welsh control site compared to our Shropshire sites. And we can then also look at zinc and selenium. So I just chose these 2 because they kind of have interesting ones for mayflies. But in general, we found that the heavy metal content was particularly low in all the sites, really. I'm wondering if anybody's recognized why the lead was high for Plecoptera. Perhaps you can have a guess in the chat if you want to. So basically, we're aware of the fact that there is heavy metals in the site. It doesn't seem to be moving into the invertebrates quite easily. We didn't find any of the europium, which is a metal that is found very commonly in the pulverized fly ash. So that hasn't been taken up by the invertebrates either. So generally it was a fairly good news story. Not so great for the MSc because it gave him a little less to talk about in his discussion. but good from the perspective of it not being that bad a site. However, because we hadn't taken any samples from the soils and from the trees and things, we thought, well, maybe we could do some more work. So last year, one of my final year students did his project, taking samples from the sediment, from leaf litter, from tree bark, and from the water itself. And we were able to run those at Harper and found that things did have metals in them, particularly iron, which again, if you want to have a guess in the chat why there's iron there, let me know. But what we were particularly interested in was that there was a difference between deciduous trees and evergreen trees. We found that the deciduous trees were more happy to actually take up metals. We're finding quite high levels of metals in the tree leaves that we found on the ground. In comparison to the evergreen trees, we find that they were quite low for metals. And so it was suggested perhaps that the trees that lose their leaves in the autumn are probably less selective about the heavy metals that they take up. But of course, those metals in the trees go into the river and then they get eaten by the invertebrates, particularly shrimp, which shred the leaves. So there was a suggestion that we might do some further work by maybe segregating the leaves into all the different species and seeing if there's one particular tree that takes up more of those metals and therefore is increasing the movement of the metals. And finally, he also looked at different pH conditions for the sediments, obviously finding as things became more acidic that the metals started to move out of the sediment. That's another pathway that could be explored a little more. Essentially, if that river becomes acidified, you might find that those metals start to move. So really what we found then, and kind of kept it short and sweet because it's now 6:40, basically there's, you know, pathways that these metals can move. It's quite variable, but thankfully our site at Crockford is not overly contaminated. And now my voice is gone. Sorry, there you go. There's some references if anybody would like to actually look things up afterwards, you're more than welcome. If there's anything that you don't get to read, just drop me an email if you're interested in having a read of them. I can send it to you by email. Thanks.
Thanks, Lucy. You've got a couple of responses in the chat on your question. I don't think
I'll just stop sharing. Okay. Ah, yes. So they are coming back. That's great. So someone just posted that the salmon have actually been making it back into the Thames. So I was pretty sure that they had been, but I don't like saying that unless I've actually read it. And it just time escaped me this afternoon. Yes, iron ore waste, but also just the actual metals in the rocks. That's so much iron. So we have quite a few plugs that have come undone lately in one part of the catchment. So you can see some of the orange water moving. And so there's a few mine shafts that basically are kind of still there and they're full of water now. And yeah, there we go. So anyone have any questions?
I'm just checking, see if there's any hands. Can't see any hands.